Hi everyone, it's Mr. Sinti. Today I'd like to discuss with you something that I think is kind of intriguing, and that is when you talk about the earliest forms of life on the Earth, the first cells of life on the planet Earth, we believe that the first genetic material, in other words, if you're familiar with the genetic material in our life, in our cells today, is DNA, we believe that the first genetic material was ribonucleic acid, RNA, and not necessarily DNA. And so this particular video will be a discussion of that and some of the reasons why we think that RNA might have been the first genetic material. And I hope you find it interesting. And so one of the first things that I want to say is that life is sort of partially defined by inheritance. In other words, the fidelity, the copying of the cell with precision, and that genetic material codes for protein. And so this is sort of where we're coming from. But as you may know today, in today's cells, just to review that concept, is that the genetic material stored as DNA is then opened, and then RNA is transcribed, and selected areas of DNA are transcribed into RNA, and RNA serves as an intermediate for the production of protein in the cytoplasm. In other words, the ribosome itself is composed of RNA, transfer RNA, and messenger RNA. And this is RNA polymerase transcribing RNA. So these three types of RNA, messenger, transfer, and rRNA, are critical in today's understanding of how proteins are made. But how is it that RNA was the original material? I mean, today, RNA is the messenger for coding for proteins and enzymes in particular. But how is it that it works out? So many researchers have proposed that the first inheritable material was RNA and not DNA. And so why do we think that? Well, because RNA can function as an enzyme is one of the strong reasons. And let me highlight that. So RNA can function as an enzyme. Again, maybe most people believe that proteins are the only type of enzymes, even though protein is a great enzyme, which is a molecule that speeds up a chemical reaction. But RNA, we now know, can act as an enzyme. And so it sort of solves the paradox of what came first, genes or enzymes. And so RNA itself can act as an enzyme. And so as it turns out, if you take RNA monomers, in other words, nucleotides, they can form connections between them. As you can see down here, you could see them linking together, forming small polynucleotide chains. And they can actually self-replicate. And so synthetically in the lab, we can actually observe this. And so polymers are added to a solution of monomers, and those can actually be copied. And especially when you add things like zinc, and zinc acts as a catalyst, and so it can actually reach like 40 nucleotides in length. So pretty impressive. So RNA is a self-replicating molecule, for one thing. And then I wanted to give a shout out for Professor Thomas Cech in the 1980s, discovered in his work with RNA that RNA can actually act as an enzyme. And there's a couple of examples of this, that ribozymes are enzymes that can remove the introns from RNA during RNA editing. So they can actually be catalytic in nature. And that ribosomes help to catalyze the synthesis of new RNA polymers as the result of that. You can also look at the large subunit of the ribosome itself, which catalyzes the formation of the peptide bond. And so in this sort of prebiotic world, because that's sort of what we're discussing, is like the early evolution of the first cell, RNA may have been fully capable of catalyzing its own replication. And now, why is that possible? And it has to do with the fact that these RNA molecules, since they're single-stranded, they don't have to be, there's only one strand. It can be double-stranded, but it has more molecular flexibility, if you will. It has more. It has a kind of a more diverse three-dimensional characteristic, which allows it to be a catalyst. And so in the lab, RNA sequences can evolve. In other words, they can sort of change. They have a genotype, which is the nitrogen base sequence. And then if that nitrogen base sequence were to change, it would manifest in a three-dimensional shape change, which is what we would refer to as phenotype. And so it has both a genotypic and phenotypic component to it. And so therefore it's capable of interacting with surrounding molecules. So there's like an interaction and a cooperation that RNA has with other molecules as a result of different nucleotide sequence and then different variant structures. So it's pretty cool. And so under particular conditions, some of these RNA sequences are more stable and they replicate fast, and then there's fewer errors than other sequences. However, that being said, that an occasional replication error would sort of, if you will, create perhaps a new structure that would be better at replication or a better catalyst. And so there you have the sort of the hallmark of biology, that through mutation or a mistake, a new variant comes into play, and therefore it's more competitive, and then it reproduces better than another one, and so therefore it has a foot up in terms of natural selection. So this sort of RNA-directed protein synthesis may have began kind of simple as just amino acids being attracted to RNA. So you get amino acids attracting to certain sequences. And then RNA would function sort of as a template, holding them together and then maybe allowing them to be linked, forming simple polypeptides. When you think about it, that's one of the functions of ribonucleic acid today and ribosomes, so ribosomal RNA in particular. And so take a look at this. Like if RNA synthesized a short polypeptide, it might actually itself behave in helping RNA replicate. So in other words, you're getting close to like today's sort of biology where RNA directs protein synthesis, and then these are enzymes which go back and do other things. And so you're starting to see that perhaps this molecular cooperation between RNA and other proteins, and this is a picture of a protein here, so proteins can help speed up the replication of RNA and therefore assist in the success of a cell. And so let's back it up a little bit and talk about these protobionts. And so these are not truly cells, but they could form through self-assembly, and they sort of embody some of the hallmarks of today's cells. And let's kick this around a little bit. So living cells may have been preceded by these protobionts, which are basically just little aggregates of abiotically produced molecules. You know, what does this mean? Well, basically these molecules are things like amino acids are in here, and you can get little nucleotides in here, like little RNA nucleotides. And again, this might be the phospholipid bilayer. And so technically these things are not living cells as we know them today, but they're made up of organic molecules that are present in today's cells, and basically they're aggregating. And so what we call these kind of structures, they have a variety of names, but in general they're called coacervates, and that means from the Latin meaning to aggregate or to assemble together or cluster. And so basically this sort of journey, if you will, to today's modern cell may have begun by a sort of sac-like phospholipid bilayer forming, enclosing water inside, and maybe it could have captured RNA inside. And then RNA acts as an enzyme itself, and again acting as a ribozyme, directing its own synthesis. And so you could actually have two identical RNA molecules, and so basic replication is starting to occur, basic metabolism, as I'll mention in a moment. And so the protobionts don't technically reproduce precisely, but they maintain sort of an internal chemical environment from the surrounding outside, and it sort of has the properties of life. It has basic metabolism. And so in the lab, droplets of these abiotic produced organic compounds assemble naturally. And this is the part that I find pretty cool. Just you put some phospholipids and some RNA and some water onto a slide, and it will just naturally form. The bilayer just naturally surrounds water inside. And if you get these molecules enclosed inside, you get something that forms what we'd call a liposome, which is basically lipids including some other molecules inside. And so once that starts happening, once you have this phospholipid in a liposome forming this bilayer, a membrane, basically simple osmosis can occur. In other words, water might have a tendency to be attracted to a more hypertonic environment toward the inside or outside. So you have movement of water, diffusion of molecules across. And then you might even have a voltage potential developing depending on the electrical charge of certain molecules. And then you have a capacity to do work, ion transfer between surrounding cells. You can have these liposomes growing inside. Maybe larger ones begin to engulf smaller one, giving birth to smaller ones. They're sort of just like advanced oil drops, these coacervates or these liposomes. But they're kind of interesting. They're sort of under the microscope, you might be fooled into thinking that is a true cell. Especially when you get into, when you look at, if you throw in enzymes into the mix, and they start getting incorporated. Like you can get simple chemical reactions. You can get starch forming from glucose, in other words, a polysaccharide, and then you can get starch hydrolysis. You can have amylase break it down into maltose, and then that can diffuse out of the cell. Phosphates can be removed out of the cell, and so simple metabolic reactions start to take place. So these probionts are capable of absorbing substrates from the outside and releasing products from catalyzed chemical reactions. It's starting to sound lifelike. And so, unlike some lab models, the protobionts that formed in the ancient seas wouldn't have had such maybe refined enzymes. Okay, so I just wanted to point that out. However, some molecules produced abiotically do have catalytic capacity. So in other words, this sort of rudimentary movement of materials across the membrane, some basic chemical reactions are visible. And so you can see, like, you can have carbohydrates being enclosed in here. You can have simple metabolic chemical reactions by coacervates. And then again, liposomes under the microscope capable of division. So pretty interesting. So in general, these protobionts, these pre-cells that had RNA inside of them, like this, okay? And so they're very simple. They have RNA inside, and they have some molecules. So basically, natural selection could refine them. And what do we mean by that? Well, just like in a population, there's going to be variation in these protobionts. And so some of these are going to be, let me see if I can illustrate that. Some of them will say, have certain qualifications for success in a given environment, and others do not. Maybe these ones are not suited for a particular way, and so these ones won't survive, and these ones will. And so you can see how through mutation of RNA and different metabolic chemical reactions, some of these cells have a preferential reproductive success. So once primitive RNA genes and their polypeptide products are packaged within a membrane, they sort of start to evolve as units. So this is different than this. And so this begins to be more successful, and therefore, as time goes by, the population will have more A than B, and that's what we mean by evolution. So the population of that particular variant will be more reproductively successful than this one. And so we can start to begin to tease through this, especially when some of the interactions between RNA and protein is what I was referring to earlier as molecular cooperation. Once you can refine that and that there's some favorable components enclosed in a membrane that's different than the surrounding environment, and this begins an exclusive cooperation that can be more reproductive than other protobionts. And so an example of that, let me give you an example so it isn't so abstract, is that if an RNA molecule ordered an amino acid into a primitive enzyme, okay, so you get this, and then that is capable of extracting energy from some sort of inorganic sulfur compound from the outside environment, then that energy for that one particular cell or pre-cell, the energy of that could actually assist in RNA replication. So by keeping the gene and its products, these enzymes, keeping the RNA and its products enclosed rather than it being outside, then it could be more competitive and therefore be more successful than other cells, and therefore, again, evolve. And so more successful protobionts would grow, and they'd split, and they'd distribute copies of their sequence and their genes to their offspring. And so this is the way biology works today. And so even if one such protobiont arose initially from an abiotic process, its descendants would vary. They all wouldn't be the same because of mutation, copying errors in RNA. And so you can continue to see how variation would proceed, and then some would be more competitive than others. So you can kind of see how perhaps, I'm throwing in perhaps, maybe, because we don't know for sure about this, but basically you could see maybe how life could have sort of bootstrapped itself up through this process. And eventually evolution, via differential reproductive success, it would vary between individuals, that have different primitive metabolic reactions and the other ones wouldn't. And then maybe, so here's the transition to DNA. Perhaps one refinement was to replace the RNA as the genetic material with DNA, which is a little bit more stable. And therefore RNA might serve as a better intermediate for which it does today. And therefore you could have certain genes being transcribed and others not. And so you get differentiation in cell, in multicellular organisms, etc. And so when did this all occur? And so again, there's debate about the origin of life. And so you cannot prove that these chemical processes actually created life on the primitive Earth. There's no way that you can prove it. These are just sort of hypotheses. These are the steps that could have happened. And so the origin of life is really subject to speculation, and there's alternative views, but that's fine. I mean, that's the nature of science too, is just investigate all alternative views, and I will certainly welcome that. And amongst this is debate whether or not these organic molecules were on Earth that were synthesized by the Earth itself, or whether they came in from meteors. I mean, maybe that's where the organic molecules were created. It's not known. And then some debate surrounds, like, where did these organic molecules come from to form these liposomes or coacervates and RNA? And some believe that they came from the deep in the ocean and these hot springs in the deep sea vents that generate a tremendous amount of energy and produce the perfect surroundings that would allow for organic molecules to be created in simple metabolic reactions. And so others believe that maybe they lived in the deep sea vent, as I was mentioning before, but then in other sites could be sometimes these hot springs are great places where simple coacervates could form because there's tremendous energy here, and there's prokaryotes and bacteria that live in here today, and they thrive in this hot water environment, and they're particularly high in iron and nickel, which help catalyze chemical reactions. And so maybe this is occurring in hot springs or deep sea vents. We don't know. However, what's fascinating about this, as our understanding of our solar system increases, we start to look beyond the Earth beyond the question, well, where did life begin on the planet Earth? We could take that same question, where did it begin, or is it occurring on other planets in the solar system? And you're like, well, what do you mean? Well, like for example, on one of the moons of Jupiter called Europa, if you're familiar with that, we know from our spaceships that are traveled to Jupiter, we know that there's ice that is covering the moon of Europa. But we also believe that underneath it there could be tremendous ocean and water, liquid water below that ice. It's not known for sure. And maybe in that liquid water there could be volcanic action, there could be deep sea vents, and maybe we'd find the precursors or life itself on other planets in the solar system. So I find that curious. We might be able to search our own heritage on a distant planet. Isn't that fascinating that we were looking for the origin of our own being by traveling outside of the planet Earth? Pretty cool. And again, Mars is worth studying as well, because though Mars is cold and it's dry and it's so-called, I'm going to go lifeless as so far as we know today, maybe it was relatively warmer and wetter before in CO2 rich atmosphere billions of years ago. And so we think that maybe Mars would be a good place to test the hypothesis of how life originated on the Earth. And maybe we could find some of these prokaryotic cells present today on the planet Mars. Certainly interesting at that. So debate about the origin of terrestrial or extraterrestrial life will abound, and I don't think I will be able to clear that up. But it's fun to be able to get into the debate, so welcome. So there is certainly a leap from aggregates of molecules, in other words, these coacervates, to even the most simple prokaryotic cell. It's immense. So I'm making it seem like it's pretty simple, just going from a protobiont to a first cell. But basically, there are steps along the way, and I hope you're able to sort of see that a little bit more clearly. So these changes must have occurred, but sort of like evolution itself, in small little incremental steps until you get to the first cell. So the point that we stop calling these membrane-enclosed liposome compartments coacervates, protobionts, and calling them living cell, it's a little fuzzy in terms of our definition. When is it the true cell, and when is it a precursor cell? It's certainly a fair question. But what we know for certain is that these prokaryotes have been flourishing on the planet Earth for 3.5 billion years, and that powerful statement, that all the lineages of life on the planet arose from those prokaryotes. So give it up for the prokaryote. All of our life on the planet Earth has arisen from these original prokaryotes. And so the tree of life, with all of its many branches, all came from those ancient prokaryotes 3.5 billion years ago. So there you have it. And so somewhere 4 billion years, when the Earth finally the crust started to solidify and started to cool down just a little bit, somewhere between 4 and 3.5 billion, we have fossil evidence of 3.5 billion. The first cell finally came into being, but we'll never know for sure how it really began. So hopefully you found that to be interesting and informative. Thanks for watching.